Hello everyone. In the last session, we were learning about the genetic codes and also we listed the 64 genetic codes and the amino acids, 20 amino acids we wrote, and the expansion of amino acids also we learned. So in this session, we are going to study about the salient features of genetic code. The salient features, this is the important one, 5 mark question it is, repeatedly they used to ask in the examination, write the salient feature of genetic code and all. So today we are going to study the salient features of genetic code. So the first salient feature is salient feature of genetic code. The salient feature of genetic code, the first one is, you might have seen in the checkerboard, I wrote you only 3 nitrogen bases. Only 3 nitrogen bases in sense like UUG, AUG, UUC, like that I wrote, right? So having only 3 nitrogen bases in one genetic code is known as, or it is known as triplets. Always they will be in a triplet form. That's why it is known as triplet codons. And how many genetic codons are there? 64 genetic codons are there. Out of 64, 61 codes for amino acid and other 3 are known as stop codons. Stop codons in sense, wherever the 3 stop codons are present, there the translation process ends. They do not further code for any amino acid. So that's why it is known as stop codon. Now the next salient feature, so that is the first salient feature of genetic code. The next is same amino acid codes more than 3 to 4 times with the genetic codes. This nature is known as the nature, like you might have seen the amino acid. The amino acid phenylalanine. This is the first amino acid we wrote in the checkerboard, right? So this phenylalanine amino acid coded with UUU triplet codon, and also it code with the UUC. So what we have to learn here, the same amino acid get coded with more than 3 to 4 more than I have written only, for example I have written 2 different genetic code, but these 2 genetic codes are coding, or these 2 genetic codes are coded with the same amino acid. If you look at that checkerboard, you can see number of repeated amino acids are present coded with the different genetic codons or triplet codons. The next is, so that is the second salient feature of genetic code. One thing you have to understand here, the nature of genetic code, like the same amino acid get coded with number of genetic code. The code is known as, the nature of the genetic codes are known as Degenerate. degenerate. Having this feature is known as degenerate feature or degenerate nature. The next is the code read for mRNA. For example, we have a strand of mRNA here, right? So, for example, if it is having the nitrogen bases on this, what we used to call these nitrogen bases, the 3 nitrogen bases, they will become a triplets. So this is one triplet codon, this is one triplet codon, this is one triplet codon, and this is one triplet codon. Now what it is doing here, the mRNA, it is ready now to code with the amino acid for the synthesis of a protein molecule. What it should do now, it should code for the amino acid here. You can see the fashion of triplet codons here. They are continuous. Can you see any like commas, any full stops after every triplet codon? No, these are in a continuous fashion without any punctuation. Okay, so this is also one feature of genetic code. If it has to synthesize if it has to synthesize any protein molecule, it should be in a it should be in a continuous manner, continuous fashion. So that fashion in your book they have mentioned like contiguous fashion. It means contiguous fashion means without any punctuation, without any comma or without any full stops. The triplet codon are there on the mRNA, or they'll read on the mRNA in the contiguous fashion. It means without any commas, without any full stops. Okay, they'll be in a continuous form. The next is the fourth one. The genetic codes, almost all genetic codes, nearly the genetic code is universal. Universal means what? It is present everywhere in the nature. It is not like that. Right from the bacteria to human, up to human, right from the bacteria, in sense right from the unicellular organism to higher organism. In every organisms, the genetic codes code for the amino acid. The same genetic codes code for the same amino acid. For example, UUU, it always code for the amino acid phenylalanine. For example, if we are talking about the bacteria living organism, if we are studying how the bacterial cell, how the protein synthesis in the bacterial cell, if we start learning about the bacteria microorganism, we used to see the nucleoid, the DNA, and then RNA, and then protein molecules. If we start learning all those things, we'll find the same answer. In bacteria also, and also in human beings. There is no change in the unicellular or multicellular organism. The process is same. The same process in sense, I'm sorry, it's not a process, it is like the same genetic code codes for the same amino acid. But here few exceptions are there. Exceptions in sense, the mitochondrial process in some organism, the mitochondrial process, and in some protozoans, the genetic code, the different genetic code codes with the different amino acid. Here the variation occurs. Only these are the exception we find in the salient feature, rather than all the living organism. In all the living organism, these kind of genetic codes with the this amino acid. For example, I have written here, if you take any genetic code from the checkerboard, whatever you see, like at the last what we have written, GCG. So the GCG genetic code codes for glycine. The same components are present in bacteria or in the human beings. So this is the genetic coding process is there, no. So that is nearly it is universal. Only exceptions are in some mitochondria and in some organisms mitochondria and in some protozoans, different genetic code with the different amino acid. And the fifth one is, the next is the genetic code. We mentioned like the stop codon we mentioned in the first character, no. The stop codons, we were talking about the 3 stop codons, right? So the 3 stop codons are UAG and then UAA, UGA. UAG, UGA, UUA. So you might have seen these triplet codons in the checkerboard itself. So UAG, UAA and UGA. So these are known as 3 stop codons, or it is also known as nonsense codons, right? And one more thing I must tell here, that is the triplet codon. So here I am going to write the 6th character, 6th salient feature. For example, AUG, its function is dual function. Dual function it has. So what is that dual function? The AUG codes for methionine, right? Let me erase this. Methionine. So what is the first function? The AUG triplet codon. This is coding with the amino acid known as methionine, and also the AUG triplet codon, it is known as the initiator codon. It is going to, wherever it is present, from there the translation process occurs. So that's why it is known as initiator codon, and also it is coding for the amino acid methionine. So these are all the 6 salient feature you have to remember in the or you have to remember, and you have to answer if they ask in the examination. The next concept is genetic codes and mutations. We are going to talk about the mutations and genetic code. So the main concept I'll describe here. The thing is, while studying the disorders in the last chapter, what is that? Sickle cell anemia, right? You might have remember that disease. We discussed about that disease. In the sickle cell anemia, what will happen here? How the disease occurs? If only the glutamate, this amino acid, it get replaced by the valine amino acid. Only because of the change or replacement in nitrogen bases, the coding differs, right? That results in the disease known as that results in the formation of or occurring of disease known as sickle cell anemia. While describing that, I used one term known as point mutation. So my concept is, today we also discussing about the point mutation and frame shift mutation or deletion mutation. The point mutation is the mutation where change occurs in single base pair or single nitrogen base. So that results in the formation of, or in the that results in the formation of diseases, right? So that disease or this change is known as, first of all, we used to take the term as mutation. All right, so only one base pair get replaced by one more base pair. Here the chain refers to addition or deletion. Like, for example, if we give one example here, I'll take this. For example, we have the 4 triplet codons here. 1, 2, 3, 4. If one base pair get deleted, what will happen? If one base pair get deleted or one base pair get added, that results, the process is known as mutation. That change occur in these base pairs or in these triplet codons are known as mutation. That results in the formation of diseases, some serious severe diseases. But here we don't know if the base pair is getting added or if the base pair is getting deleted, whatever the change is. But if any kind of change occur with one single base pair, it is known as point mutation. And one more thing is present. Here if they ask to write, let me tell you one more thing here. If they ask to write, give one example for point mutation, what you will write? You have to write about the sickle cell anemia. How the disease causes? The disease causes but only because of the deletion or the replacement of what? The amino acid. How the amino acid get replaced? The nitrogen base. The nitrogen base, instead of A, the U nitrogen base get replaced. That's why the disease occurs. Here we were talking about the same thing, only one single base pair, because of the replacement of one single base pair results in the formation of disease. The same thing we are talking about that is nothing but point mutation. So one more thing here, if they ask to write the example, you have to write the sickle cell anemia and you have to write the amino acid name also. Glutamate is replaced by valine in the beta globin chain. That also you have to, in the beta globin chain, the gene or the base pair get replaced. So because of the change takes place in the base pair, the change takes place, and in the coding amino acid, the amino acid also get changed. Instead of glutamate, the valine amino acid code for the code in this beta globin chain. So that is one example for point mutation. So the second one is frame shift mutation. In the frame shift mutation, the change occur in one or two base pairs. That results in the formation of some kind of diseases, and the process is known as frame shift mutation. So you can see the difference between these two. Point is only one base pair is getting changed. The frame shift mutation is one or two base pairs getting changed. Either it is getting added, or it is either any other base pair is getting added in the gene, or two base pairs getting deleted, or it is changing its place. So anything will happen, anything will occur. So these mutations are known as point and frame shift mutation. So with this we will end today's session. In the next session we will be discussing about the translation. We are going to begin the process known as translation. Thank you.